Hi guys, I'm here with the research group from Haines City, Florida. We have Dr. Yadiel, intern Vanessa, intern Vanessa, and Dr. Lee, and I am Professor Reed. I sit in on research groups all over the world to see how their scientific process is going with finding new experiments.
Is the camera on? Oh, hey! My name is Dr. Yadiel. I'm here with Dr. Georgia, intern Vanessa, and Dr. Lee. Today we're gonna be discussing. This is a bunch of hogwash. How are we gonna get the people interested in science again? No one's gonna listen to this. No one cares. What are we gonna do?
You know what people do, Karen?
What?
Existing. We should look at like one of the basic things in our existence.
What are you talking about?
Dunno.
Dana.
It's pronounced DNA, Dr. Lee.
I think Dunno sounds better.
What's Dana?
DNA is the genetic code of every human being. It's actually pronounced DNA.
Dunno.
Okay.
And one of the things that has been tested through Meselson and Stahl was the replication of this genetic code.
Dunno.
Wait, wasn't it the part where they had like 3 different types of replications?
There was 3 theories going around with Meselson and Stahl.
Yeah, a couple silly ones. And one that works.
Okay.
There was a few silly ones.
And what are those 3?
So the 3 theories were conservative, dispersive, and semi-conservative. Meselson and Stahl found out that DNA did in fact replicate through semi-conservative, which means that one of the reoccurring daughter strands of the DNA did have an exact replica of the parental DNA.
How did they find that out?
Well, they found it out by observing some E. coli, and I have this pretty nifty diagram here.
Do you need help with that, Miss?
Yes, intern Vanessa. Alright, so what they found, that here's our DNA strand, and this DNA strand is being separated by this helicase right here, which moves in a 5' to 3' direction by splitting the hydrogen bonds that are holding the parental strands together. Then, as it replicates, the DNA polymerase puts new nucleotides that are the matching complementary pair of the parental DNA as the DNA gets split. And thus, when it's completely split together, you have 2 exact strands of DNA, with one strand being the new synthesized DNA and one strand of the DNA being the parental strand. Conservative was this wish-wash about how the parental strand just replicates and then it has a different new not identical strand of DNA, and dispersion was where the 2 DNA strands were mixed together.
Wasn't it one where it says each strand of both daughter molecules contains a mixture of old and newly synthesized DNA?
And the conservative was in which like the old one was replicated, and then you would have the old DNA still there, and then the new DNA was just some replication that was completely new DNA.
I don't know. I kind of like the conservative theorem. That sounds pretty convincing.
Why?
Well, I mean, think about it. Well, why would the cell go out of its way to try and make new cell, to try and make new DNA synthesis using parts of the old one? What if the old one is just old and grody, and then unusable? What if it's too rusty? What if it just doesn't work?
Then the cell performs apoptosis and dies, just like your ideas should.
Go on.
All right, and that was day 1 of our study. We're gonna work on some more study in day 2 and 3. Stick around for the next episode.
Hi guys, I will be following these three scientists and these two scientists and an intern Vanessa. I will be following them through their journey of figuring out Meselson and Stahl's idea of semi-conservative DNA replication.
We're doing test subjects.
Hi guys, day 2 of being with our research scientist from Haines City, Florida. We have Dr. Oh God. Okay, guys, scientist is a collaborative effort, and we don't need to do this to people who are just a little bit messed up in their ways.
I literally just said I thought it was conservative theorem.
We just have to explain it to them that they're wrong.
Doesn't always work.
And then we provide evidence.
Well,
So
he
much
was a,
less fun.
but he was a great test subject.
Intern Vanessa, please.
For science, I swear.
So, have you gotten your research from the other research team I hooked you up with?
I've literally been tied up and put a blanket over my head. What research have I done?
Have you gotten the research?
Yeah, they forwarded it to me.
They
Put it in
forwarded it
spam.
to you? You put it in your spam box?
Yeah, it was tasty.
Okay.
Of course.
So, have you guys updated any of your models yet to match? Yes?
Yes. 50% copy, just like semi-conservative.
What about you, intern?
I didn't really.
Go on.
And I found out that in the diagrams of conservative and dispersion, this, I can't say word right.
Dispersive.
Dispersive. Dr. Yai.
Intern, you're close to being fired.
I mean, you were close to being dead. So.
There was some complications in it. For
Explain.
I was gonna say explain.
Relax, just speak, my darling.
I prefer that you don't touch the intern.
What kind of
Thank
mistake?
you. I don't think so. Well, in conservative, it wouldn't not make sense if there would be multiple replication of the parent DNA. And if it was true, then the DNA will get smaller and smaller and smaller because, well, or there will be too much and space will overflow. In dispersion, there was too many complications in it.
Okay. Thank you, intern Vanessa, for your insightful thoughts. Alright, so, well, that was day 2 of this wonderful collaborative scientific group that needs to get along.
Get along! We need to get along!
along.
Can you always call me by my real name?
Intern Vanessa, please. We don't have time for this.
Alright, now, this is gonna be day 1 of my experiment. I'm gonna think of an experiment to do, where I support the conservative theorem because they don't believe me. They think I'm crazy. I'll show them. I'll prove them wrong. Now you see here. This is my theory that I came up with. Now see, when a DNA splices, the conservative theorem allows it to create a totally new DNA that lets it age freely, the same way that it does with cell division, or the same way that it does with its own replication of RNA. Now the reason why I think that semi-conservative doesn't work is because there wouldn't be any reason for the cell to reuse old strands like this. This is really weird, but they insist that I'm wrong. I'll show them.
Hey guys, day 56 of this crazy team of scientists in Haines City, Florida. They've been such a joy to work with so far. So, guys, how's creating your experiment going?
Going great. I'm just rolling out my plan to use technology to recreate the experiment
What about you, intern?
Well, I've been trying to disprove your theories, Dr. Yagi, and I still see no proof.
That's well.
That's,
Don't you have some papers to write?
yeah, you're not supposed to be doing science. You're the intern. Go file those documents.
Okay.
Bloody bomb.
So, guys, do you have a detailed plan yet?
Oh, I have one, but it's a surprise. All I'm gonna say is it's gonna involve some N15, N14, and
Some test tubes?
maybe some lab states too, of course.
You better tie your hair back.
Okay, all right. Apart from that, it's gonna be a surprise. You all certainly will be surprised.
I'm using this technology that I was recently given to test it out to see if it still follows through with Mendel. Downloaded it off the internet. Said it was free and virus free.
Small rebellion.
I like working with these scientists. Thank you. That was day 56.
Alright, okay. So, day 2 of my experiment. Alright, so what I have here is I have a solution of N16. And what N16 is it's full of bacteria right now. And what I'm going to do is I'm going to have the DNA in this bacteria replicate for 1 to 3 cycles. 1 to 3 generations. They're gonna replicate 3 times. I'm gonna try and prove my experiment. What I'm gonna do is I'm gonna have the different bacteria replicate into N14 and then N15, and after that I'm gonna put them in a centrifuge of cesium chloride, and then I'm gonna put it in the centrifuge and see what happens after there. I'm gonna use this experiment that Meselson and Stahl did in order to prove my experiment. All right. So. Bacteria. Okay. Put the bacteria in. We'll see what happens. Day 2 of my experiment. Unfortunately, the second cycle started. It didn't exactly prove my theorem yet. The DNA densities were intermediate, which is not what my theorem necessarily supports. My conservative theorem supports that all heavy or light densities of the DNA are there, but no intermediate. Must have been some sort of limitation I did, but I'm still certain that there has to be some sort of way to prove this wrong. The semi-conservative theorem still hasn't been proven, so there's no reason why we shouldn't reject the null hypothesis just yet. Well, we'll see what happens. So, final day of my experiment. I did get some good news. that the dispersive theorem of the second cycle, or the third cycle, I'm sorry. The third cycle of the reproduction. I think the dispersive cycle was ruled out because it says the third cycle said the two intermediate DNAs were there. That's not true. So at least I got that. There's no evidence that the semi-conservative theorem was ruled out, and the only thing I have is that mine. Screw this.
Hey guys, did you leave all the lights on?
No.
Huh, that's funny. I remember turning the lights off.
The Kool-Aid.
Ooh.
Drinking contest, go.
Proper lab safety. No drinking in the lab or touching anything with your senses. Alright guys, you're fine. Get up.
That was my ploy the whole time! My goal wasn't to actually prove Mesel's stupid theory. That wasn't cesium chloride, it was actually cyanide! I'm gonna go get McDonald's.
That's great. Guess that's a wrap.
Note to self, I'm never coming back to Haines City to do another research group. Well, that's the end of this experiment log for a while. Not coming back to Haines City. Maybe I'll move to Russia. They have some better scientists.